It must have been him, just a glimpse of a face in the crowd. That's all it takes. My heart is racing. I stand in a trance as I stare at the back of a bus, knowing there's no use to.
So in a previous session of the lecture, we looked at some from the classical genetics point of view. We went through some of the necessary terminologies to define and also understand the basic of our concepts of genes and the outcome of the different gene interactions. Okay. Now, here from the aspect of molecular genetics, which is the core of the modern genetics, how do we define the genes? So from there are two different versions of the definition of the gene from the molecular genetics aspect. One is a rather narrow definition, is under which it states what gene is. The segment of the DNA, along with the whole DNA and genome, on a small area, some part of the area which carries the information on for protein synthesis. So the gene equals the area that code for protein synthesis is the more direct and kind of intuitive definition of the gene, because when it comes to actual real the role of the genes are for making proteins after all. So that's kind of a well received the general definition of the genes. However, this is not 100% entirely accurate definition, because not all genes are directly related with the proteins. There are some non-protein coding genes. Then how do you define those? As they are non-genes, but nonetheless they are also the genes. So with this narrow definition of genes that only concerns the protein synthesis information, we cannot handle such example. But anyway, let's take a look at this, the narrow definition of genes, why those are relevant in that. So here this diagram once again showing the connection between DNA and the protein, in that in the middle there's an RNA, which is the mediator of this delivery of DNA information into fruition. So that here the terminology, the codon, which we are going to take a look at later on this chapter. The DNA sequence here, whatever they are, however random they might look, but it's combination of DNA sequences. The meaning of this combination of DNA sequences is where certain specific combination of a particular DNA sequence can now signify a specific amino acid. So the protein, if you think about the protein, another name for the polypeptide is another way of calling protein. Sometimes it's called the polypeptide. So the protein is made of all these different types of amino acid assembly. Then how each different amino acid, so who determines? If you look at the protein and under the basis of protein is like assembly of different amino acids. So now what it comes to, what matters is now then what's the order of the different amino acids, and position, a certain position, what type of amino acid comes, and the next position, which type of amino acid come. Then they are connected. Then as a end result is different proteins. The unique protein is actually a result of a unique such combination and assembly of different amino acids in different orders. Then at certain position, what type of amino acid, who decides? And DNA decides, like as I preach. And how? Because DNA sequence at a certain position, like here as you can see, 3 particular packet of different type of DNA sequence temporarily delivered into in the form of RNA, and this RNA will have some specific relationship and specific particular RNA nucleotide sequence is able to define a specific amino acid, and that's how DNA, certain sequences, certain order of DNA can overall determine the protein. So now we are talking about the gene as we define is only certain small segments of your entire DNA has this type of meaningful relationship here, and then we call that as a gene. Even though in this diagram the another gene, gene 2, immediately precedes the gene 3, which shows this relationship, but in reality we know that I have already shown that things are not like that. Usually a whole different vast majority of DNA chromosome, only some portion here, so this is one gene, and some space, and then another gene. That's usually the case, and another gene is over here. So then each genes is separated by a huge space, meaningless space, and then next gene comes is the way your genome is organized. So now in narrow definition, each gene should be able to directly define any kind of protein information. But as I say, not all the genes are actually doing that. Some genes are not directly related with any protein coding information, but still they are part of the genes. So in order to accommodate such genes that are non-protein coding, so here the much broader definition of a gene should be corrected as the gene is any segment of DNA that can is very important. Segment of the DNA that makes DNA, but it's potentially, that's as long as any segment of DNA is capable of, has a potential to make RNA. By the way, the activity of making RNA from DNA, any DNA given region, this activity is called transcription. So transcription is nothing but actually the copying process, the complementary copy. So the RNA generated as a result of this transcription, the activity of transcription is the characteristic is it is complementary copy of either one of the 2 DNA strands. So if DNA strand is over there, 2 DNA strand, RNA is being copied starting from over here and over here. This segment will be copied into the RNA, and the practical copying procedure is by assembling the order in complement according to the complementary base pairing rule of any existing DNA sequence, and that's how you generate the RNA. And now the only question is which of the 2, top and bottom, which of the two DNA strands will be copied is the question. Okay, but as long as either one of the two is being copied, now the transcription activity is accomplished. So anyway, any area that is capable of doing, which means it has an ability, but that doesn't mean that it will always do this 24/7. No. If I feel like to, I will do. But if I don't feel like to, I don't have to do it. Is the thing that here just try to define things. So as long as this area is capable of generating this complementary copy of an RNA, then it is the legitimate. By definition, legitimate gene area is what the more narrow definition of the genes. So what kind of RNA is why the original reason why this broad definition of the gene has to be introduced is because of there are different kind of RNA existing in the cell. So we have already looked at 3 different types of RNA, including this messenger RNA, mRNA. mRNA's characteristic is a coding RNA. Why it is called a coding RNA is because the whole area of a messenger RNA, I wouldn't say this whole RNA, it's the majority of the part of this messenger RNA is directly related to the amino acid sequence. So in that regard, it is called the coding, because the sequence of a messenger RNA is the codes for specific amino acid. So in other words, it can be said of the one that specifying amino acid as a unit of codon. So the meaning of a unit of codon we are going to take a look at later on this chapter. So when it signify or when it designate a specific amino acid, it does so in a specific group, and that specific group is the unit of the group is a 3 nucleotide, and that such 3 nucleotide we define as a codon is what it is. So that's messenger RNA. So in other words, if you read the messenger RNA sequence, then you have very good idea about what type of amino acid will be assembled. If you follow this messenger RNA and what type of amino acid will be connected to result in any given particular kind of protein is what those relationship between messenger RNA and the protein. So that's why messenger RNA is called the coding RNA, right? But that's it. So much. So the messenger RNA is the only coding RNA, and the rest of the other kinds of RNAs are all non-coding RNA. So that includes ribosomal RNA. The job of ribosomal RNA is as a non-coding RNA. It does not participate directly in protein coding, but still in protein actual making of a protein, the RNA, ribosomal RNA is essential. Without the ribosomal RNA, there is no way you can make protein. Why? Because this ribosomal RNA, if we imagine the ribosome structure as this big head and a small body, so this is a large subunit of ribosome, and this is a small subunit of ribosome. Then each subunit, small or large, consists of several kinds of ribosomal RNA. In the case of a large subunit of ribosomal RNA of a eukaryote set, 3 different ribosomal RNAs, which you don't have to worry about which particular ribosome, but anyway, several different kinds of ribosomal RNA mixed with something like many different ribosomal protein many, something like a 30 or 40 different protein, ribosomal protein collectively called. So this, and in the case of a small subunit, one ribosomal RNA different from the three, those previously mentioned, the three different ribosomal RNA, and one this ribosomal RNA plus another something like about very different protein. So that's how assemble the ribosome. And the significance of this ribosome is why this ribosome is the location of protein synthesis. So without making the ribosome, there is no way you can do this protein synthesis activity. No matter how many messenger RNA you generate, then without ribosomal RNA, those messenger RNAs become obsolete, useless. So that's the reason. And in order to generate this correct ribosomal RNA, ribosome structure, and the ribosomal generating ribosomal RNA is essential, the prerequisite. And so that's the reason why it is very critical and then indispensable component of protein synthesis. And another thing is this. Actually, this ribosomal RNA particular ribosomal RNA present in this ribosome is the one responsible for the catalytic activity, which is essential and critical in connecting two different amino acid. So without this ribosomal RNA, the enzymatic catalytic activity of building proteins is impossible. So the ribosomal RNA is extremely important. The only thing is it itself does not directly reveal any the amino acid order sequence information, but nonetheless it is extremely important. So this is showing some of the secondary structure, and once you have generate any secondary structure, that means automatically this will fall into some 3D structure. And whenever you are capable of forming this type of a unique 3D structure, that means you are potentially you can act as any kind of enzymes, right? So that's where this ribozyme ability comes. So ribosomal RNAs are entirely capable of performing this function of enzymatic function, is the message. And on the other hand, the transfer RNA is the carrier RNA. So it is once again on a non-coding RNA, because this RNA itself, after being generated as a result of a transcription, the RNA sequence itself has nothing to do with any the protein coding information. But nonetheless, without you can easily imagine that without transfer RNA, you cannot perform the protein synthesis, because these are the one actually individual different amino acids being recognized and then carried over to the site of the protein synthesis, which is the ribosome. So, and the 2D structure roughly of the tRNA appears to be, looks like this. So this is another example of such a secondary structure, which is maintained by the internal, the complementary base pairing, which I said this as a intra-strand complementary base pairing. I just introduced and before the midterm exam. And one end here, now you get to see the example that we went over, this 5' over here. So this is the first end, the first nucleotide, where you can find the first nucleotide of this particular transfer for RNA strand, and this the very 3' end is the last nucleotide of this transfer RNA, and at the end of this 3' end is the place where you have this particular amino acid is being attached through some specific chemical bond establishment. So here is in each different transfer RNA can pick up a unique. There are 20 different types of amino acids. In other words, at least you should have 20 different, at least minimum 20 different types of a transfer RNA you should carry in your cell to take care of this. All each different amino acids are recognized and being brought over to the ribosome. Okay, so that's, and so those 3 RNAs are the one that initially introduced to you before the midterm exam, and now there are 3 more the identities of RNA which previously was not introduced, because those 3 new RNAs are their existence are relatively actually later recognized. Even when I was in the graduate school as a pursuing as a doctoral degree to become a PhD, the existence of these 3 different new types of RNAs were like largely unknown. Nobody actually, almost nobody knew there is such a thing like this new type of RNAs. So these are very recently recognized and characterized, and then turned out that these newly identified RNAs are also very equally important in terms of their functions, important as those 3 more classical RNAs that we used to know. So these are, and one is called the microRNA and abbreviated as a miRNA, like this. These microRNAs are, once again, this is non-coding RNA, and one interesting property of this microRNA is, even though they are non-coding RNA, but overall the sequence of microRNAs are like very much strikingly resembles. There are some kind of counterparts of the messenger RNA. So one can easily fooled by like, oh, this must be some type of the messenger RNA, and then later on, if you examine carefully, it is not actually messenger RNA, but it's a non-coding. It does not carry on any real protein coding information, but it does appear to as if it is carrying a protein coding information. It's a very interesting characteristics. But why? Why do they have such kind of a decoy? Is because I will just stop over here, because if we will move further deeper, it becomes more complicated. So it is not for everyone. For only those who are interested in the major in life science can dig in more about these characteristics, because it is a up and coming kind of RNA, and their significance in every day. Their significance is like updated more and more. But anyway, the bottom line of a function of this microRNA is control of gene expression. What's gene expression? Gene expression is the activity of transcription, making RNA. Is what we usually define. So when someone refers to, okay, this gene must be expressed, what do you mean by that? That specifically saying that this segment of the gene must be copied into RNA through the activity of transcription, and that's what it means with gene expression. Okay. So this gene expression activity is actually controlled by somebody, There are many different factors. There are many different factors that influences of this relative activity, relative strength of any particular gene expression ability, and this is one such major factor that, as a consequence of this microRNA, certain genes can switch on, turn on. That means gene expression becomes active, or switched off, turned off, and the expression of this gene expression, the activity of gene expression is going to be toned down, or even in some cases completely shut down. Okay, so it has a disability, and cells utilize this microRNA very heavily in many different types of this gene expression control. But still, this microRNA is not the only one actually which is engaged in this gene expression control. There are many different protein factors as well, are also all are involved in the particular gene expression. It's very complicated business over here. And the reason why they can do this gene expression control is overall their RNA sequence is complementary to the specific target genes. That's why you never know. We don't actually, the way you discover any particular microRNA is from the DNA region. DNA. And let's say this bottom strand will be copied into. So the complementary to this bottom strand is made in the form of microRNA. But usually the way you identify this existence of the microRNA is by examining the entire area of this DNA, and you never know which of the two is the one actually this gene product. And if you look at, then if this one is the one copied, and this sequence, the upper sequence, would be complementary to this, right? So that, oh, this region is very similar to the area that I used to know as a certain gene, is the specific relationship between messenger RNA, any given messenger RNA gene, and this microRNA gene. And that is being utilized actually by the cell to have this specific complementary base pairing relationship with a given gene product. And by having this complementary pairing, once you have this complementary pairing, then you specifically destruct this particular messenger RNA, and that's how you control the gene expression. And this lady is one of the top experts in this microRNA research, as the Korean scientist, one of the leading, very influential scientist who is now working at the Seoul National University, is one of the very leading the expert in this microRNA research. And another type of such non-coding RNA is called the small nuclear RNA, abbreviated as snRNA. And this RNA is used in the process by which the introns of the messenger RNA is being removed. So for those who have never taken any previous information about this business before, now just what's intron? So yeah, don't you worry, we will get there in this chapter later on. So there are some segments that need to be removed during the process of this messenger RNA in the eukaryotic cell, and those jobs of editing of messenger RNA is done by this small nuclear RNA and the associated proteins. So this diagram, let's not go over this. Only will give you any kind of unnecessary, complicated confusion. I guess it's just a suffice to say that there is a particular type of RNA, non-coding RNA, that is needed to do this particular activity. We call this a intron removal. And another type of such RNA is called the SNO RNA, standing for the small nucleolar RNAs. So nucleolus versus nucleus. Nucleolus is in the nucleus. There is a certain specific area under the light microscope. Usually, this specific area of the nucleus is stained very dark, and this is nucleolus, right? And what's the kind of relevance of this in function-wise nucleolus? Here, in this nucleolus, you get to see this particular RNA. Ribosomal RNA are made so in other words, this is a part of your chromosome. So those chromosomes where the ribosomal RNA making genes are clustered there. They live together in a very narrow region. And also in this area, actually the ribosome assembly. Even though ribosome assembly takes the protein assembly, because many different proteins are needed to assemble, whether it's a small subunit or a larger subunit, right? So in this subunit scale, small subunit ribosome and large subunit respectively, they are assembled in this nucleolus, and then each subunit is exported out of the nucleus to the cytoplasm. Then now they combined to start off this protein synthesis business. So basically these ribosomal so that's why this SNO RNA is called the small nucleolar RNA, because their function is the ribosomal RNA processing. And where can you find this ribosomal RNA? Just nucleolus. That's why. But more importantly, so with all these different types of RNA making, each of which is legitimately area of gene, but the majority of the area on your chromosomes are not involved in any of this RNA making activity at all, so they are non-genes. And here, the important concept. So those areas usually tend to be organized in certain heterochromatin regions. So what's heterochromatin? First, chromatin. Do you still remember the chromatin? Is whether you have in your chromosome either exist in this loosely organized, like untangled and less condensed entity, or highly condensed. When these are highly condensed like this is a chromosome, or you can pull and make all this compact, highly compact condensed area into much less condensed area. Now we call it as a chromatin. So this organization actually move back and forth depending on your need. So even the same area of your genome, sometimes it can exist as a highly condensed chromosome, and sometimes, but what do you mean sometimes? Actually, more to be more specific, only just right before the cell division. Only when this cell is about to split into two, then this whole your chromosome gets to organized into highly condensed form of chromosome. So for the rest of the period, to the aspect of the cell, if you consider it as the entire lifespan of any single cell, the majority of these cells exist, their DNA exists in the form of this chromatin, less condensed form. So that's the distinction. However, now the new terms, the heterochromatin, what is this? Versus, then there must be some kind of counterpart. Yeah, the counterpart term is called the euchromatin. So this heterochromatin is more condensed. Not as much as the chromosome, but for any chromatin, which we expect as a much less condensed, then heterochromatin structure is like a rather more condensed structure, whereas this euchromatin is less condensed. So in other words, there are some kind of a degree of a difference in condensation among the whole general area of a chromatin. Just because you are now organized as a chromatin doesn't mean that they are like uniformly, like their degree of condensation is equal. No, don't expect that. So from the overview of a chromosome, like if this is a single chromosome, a single highly condensed chromosome, then you have a certain area and a central area that labeled as a centromere, where through which actually the spindle fiber during the chromosome separation during the cell division, the spindle fiber that carry on, hook up and carry on, and then split, separate the chromosome, that we have seen this microtubule that one of the cytoskeleton protein need to be attached in certain area of a centromere and then pull this chromosome, and that's how this particular chromosome is being dragged into a specific direction. But this is the area where we call the centromere. So the central area, even though depending on what type of chromosome, the actual exact location of a centromere could be different. So certain chromosome has this type of a shape, so where the centromere is elevated up in terms of its balance, and some other chromosome, different type of chromosome, the centromere location is kind of relatively equal, like well balanced in the middle, and another centromere could be in like the other area. Like all different chromosome has a different location of centromere. Anyway, all this centromere has a specific structure and a specific function. So if this was called centromere, then we have another kind of area, like end area, the edge. The edge area of a chromosome we call this a telomere, the end of a chromosome. Remember, the eukaryotic chromosome is linear, unlike prokaryotic DNA, which is a circular. So being linear, you have these two ends. Inevitably, you have two ends of this long line, and these two ends are called telomere. So even though you condense this whole entire structure, still the telomere, the same telomere exists and ends up in the two edge of this chromosome. Then we call it as a telomere. Yeah. So here the reason why I brought this cartoon, this diagram, is to illustrate the relative association of the chromosome organization. So centromere and telomeres are generally usually organized, even though these areas are organized, not organized in chromosome, but rather organized in the less condensed chromatin. And this centromere area or and telomere areas are usually tend to form a heterochromatin, which means highly more condensed. And why only those area? I mentioned in this lecture, it was mentioned that those area, the non-gene area, right? Area usually tend to form as a heterochromatin. Why? For the one thing, if this relationship holds true, in other words, this telomere area and centromere areas are the area where you don't expect to see any of the genes, and that is the case. If you look up any area, centromere or telomere and chromosome, then you seldom find any meaningful genes. Usually you don't get to see, although I'm not saying there are not zero, nip, nada genes are there, but there are some genes exist, but in comparison of the other area, this area, the number of genes present in those areas are very, very, very scarce. And similarly, along with this your entire chromosome, let's say this area is a non-gene, let's say, then you usually, the chances are this area is usually usually preferred, tend to organized into the heterochromatin. So here the point that I'm trying to make is the specific relationship between the particular gene activity, gene expression activity, and their chromosomal organization status. The more condensed you make your area of chromatin, the more you condense, then the less likely you will be able to induce any the gene expression activity over there. Why? It's because the more compactly organized, and it's much more difficult for any those material factors that are needed for gene expression. So you thought, okay, if this was the gene, oh, this is the gene, which means the RNA can be made. Let's say this is the starting point. From here, the RNA will be generated until this area. So this is the area of the gene. So to start off this RNA making activity, RNA is being made, then what kind of actual factors are needed? Can it start this RNA making activity on its own without getting help from anybody else? No, that is not the case. So to start off this thing, first you need to have any particular enzyme responsible for this catalytic activity, the RNA polymerase. Just like if you want to replicate your entire DNA, then the enzyme, the DNA polymerase, need to be there. Same thing. If you want to make this particular area, the like RNA copying activity, then the RNA polymerase need to have this catalytic activity. But and that's not it. Not only this RNA polymerase and all kind of other factors must be involved, and all together this entire process can start and then efficiently can be processed. So to do so, all those factors are the proteins. All these protein factors needed for this RNA expression have an need to get access to this particular area. And if you imagine, like, if this area is highly compact, then how can all these protein factors can correctly recognize that particular area and then have some kind of access? No, it is very difficult. That's why there is a very specific correlation between the degree of, I should say it is a negative correlation. Yeah, the more condensed and less gene expression, because of this accessibility issue. So if you want to make this particular area highly active in gene expression, then you better make that area. So if you want to have this high gene expression, activity, then you need to have this area, this particular area is a euchromatin area, which means much less condensed area. So that is always a specific relationship. So anyway, that we are going to revisit that relationship shortly after taking care of this. But once again, another thing is there are intrinsically the heterochromatin areas. So one thing is, on a given area, it can the area of the degree of condensation can be dynamic. But another thing is the intrinsically chromatic area on your genome. That means there are in your original DNA this area, okay, this area, there are no genes. No genes live here. Then usually, like almost all the time, this area will then exist in the form of a heterochromatin, right? Because there is no incentives for you to make this area much less condensed. Why? Because there are no genes. I don't need to worry about any. So that's the relationship exists between this no gene area on your chromosome and the status of a chromatin condensation is like hetero, so that they are relatively rather more tightly, more condensed. And surprise, surprise, surprise. And then if you examine the DNA sequence of this heterochromatin region, the intrinsically heterochromatin region, where there are no genes, actually, virtually no genes present, and then it turns out that the sequence of variation of that area among different individuals is much, much, much higher than the area, the occupied by many meaningful genes. Why is that so? Why, in particular, this intrinsic heterochromatin areas DNA sequence variation among different individuals are so high, and those areas that where more genes are located, the DNA sequence variation among different individuals are much less. Why? That's, if you think about it, that's actually very natural. Let's say there is an area where your hemoglobin gene is there, and some other genes are the very important gene. So this is a gene heavy area. Let's say the sequence variations occur random. Is random, right? The DNA sequence variation is random. It will occur spontaneously as a result of random mutation. But even so, if any DNA sequence variation occurring in this gene area, any alteration of the gene sequence, the functional gene sequence, would result in some kind of a malfunction, so defect in the original gene function. So any DNA mutation occurring on this your gene area will result in very bad result. So deleterious. So bad impact. So individual will usually die. Any mutation. Yeah, it's very unfortunate. Even though whole area, entire your area, there's no gene area, so mutation can equally happen, but particularly those area that receive this DNA mutation, those individual carrying this mutation will not be able to make their lives, or even they do so, but the chances are they produce their viable offspring is extremely low. So along with the long history of evolution, those individuals carrying the mutation in their gene area will be selected out, and those individuals who have the mutation rather on this non-gene area will not have any such bad influences, so will be able to survive. So these trends will then eventually be extremely skewed into such that nowadays, if you examine, wow, how come we have so much variation on this non-gene area, heterochromatin area, whereas this area where the heavy genes densities are there, then you don't really get to see any too much variation, because those who have those variations are all no longer exist. Their offsprings are also no longer exist. That's the reason why. So this DNA variation, we say all these different individuals, their DNA sequence is very unique. But once again, if you think about it, in a flip side, if you expect that the DNA sequence of your hemoglobin gene is so unique that it will be very much different from that of the other, your friend hemoglobin gene, then you are vastly mistaken, because either one of you then cannot survive with such extreme variation. So this unique DNA sequence variation, we only are talking about those areas where there is no genes are present. So that's why this heterochromatin, intrinsic heterochromatin area, tend to have much more genetic variation, DNA sequence variation in each different individual. You think about it, like even On average, human and chimpanzees, the DNA sequence identity is more than 98%, right? So then how can you expect the whole this unique DNA sequence variation among each different individual? So you do the math. And that all those majority of the DNA sequence variation among different individuals actually come from only very limited area of the intrinsic heterochromatin area. So the reason why I'm elaborating this concept is because now we can use those highly variable area in doing this so-called DNA profiling. Two particularly well-known such area we recognized as one is a satellite DNA and the other one is a VNTR. I'm just throwing these two terms just because these two terminologies are frequently mentioned in everyday our lives in when it comes to the paying attention to this business of DNA profiling. These two satellite DNA, VNTR, whatever they are, frequently mentioned. And so without even bothering to know what they are exactly, these two different segments of DNA refers to some of highly variable area of the DNA in terms of the DNA sequence. Especially another interesting aspect about this variation is this variation not only do they come in the form of the actual DNA sequence variation, but especially in this particular whatever it is, the VNTR, is achieved through the number of repeat. What varies in different individuals is this number of the repeats. In other words, in this area, very small repeat. So each block, red block, is a particular repeat pattern of something like, hey, DNA AAG, AC, AG, CT, whatever. This particular sequence is repeated, present multiple times in very short area, very small area of this. This is a repeat. And the thing is, the individual A, person A, and this person B, the same number of repeats is different. So here 1, 2, 3, 4, 5, 6, 7, 8, 7. Whereas this individual B, exact same sequence of repeat on the same locus, is having only 4. So all this individual and this VNTR is all over the different 23 different chromosomes. So you add up and all this different number of different repeats, the variation is so complicated to the point of by counting different or repeats along those 23 different chromosomes, you can actually make the case of identifying one very unique individual, and that's the basis of safely performing this so-called the business of DNA profiling. So before we actually jump into more, then immediately one might ask, then why we have such a non-essential DNA, which is very very vulnerable or prone to this easy sequence variation? We do still carry this area because, after all, they don't have any function. No genes are there, and in the course of this whole long time of evolution, why do you still keep those stupid garbage waste area? Good question. I don't know, but it is what it is. we do still carry such a huge area of useless, repeat rich area. So the only thing that we can come up with, instead of trying to figure out, okay, they are really, really necessary, maybe the best way to answer such question is, how about we eliminate all this, get rid of all those garbage area, and then see what happens to that individual. And but that is ethically impossible to perform and find out the experiment. So the only other thing that we can do is there any other usage we can like with this area? Yes, we do, and that DNA profiling is the answer. Um, the About the DNA profiling, we actually have a little more in detail that need to be covered. So it's been already over an hour. So I guess I will just put in the rest of the stuff of the DNA profiling discussion off into the next section of the lecture. So for now, I will stop here, and then in the next lecture, the more details about the method of the DNA profiling and principle and the application will be followed.